In Chapter 4, we're going to be talking about cellular metabolism. So the first thing we need to do is define metabolism. And metabolism refers to the sum of all chemical reactions that are occurring in the body. Now, you may have heard the term metabolism before, and you probably think of metabolism as being like how quickly your body breaks things down. You've probably heard someone say, I have fast metabolism or I have slow metabolism. But we need to be sure that we understand that when in A&P, when we talk about metabolism, we are talking about all of the different chemical reactions that occur in the body. So we can talk about overall body metabolism. We can also talk about cellular metabolism, and that's really what this chapter is going to focus on. So cellular metabolism is the sum of all chemical reactions that are occurring inside of an individual cell. Now, these metabolic reactions typically occur in pathways or cycles, and we will look at some of those pathways in just a little bit. Now, we've already said that metabolism is referring to all of the chemical reactions. And so now let's break down our two big categories of chemical reactions that can occur inside of cells. These two types of metabolic reactions are called anabolic and catabolic reactions. So first let's talk about anabolic reactions. Anabolic reactions are chemical reactions in which small molecules are built into larger molecules. We've already talked about a number of these types of chemical reactions in our previous chapter. If you remember, we talked about a bunch of different types of monomers that could be hooked together to form polymers or macromolecules. And some examples of those that we've already talked a little bit about are amino acids being hooked together to form proteins, monosaccharides being hooked together to form more complex carbohydrates, also known as polysaccharides. And we also talked about little individual units called nucleotides being hooked together to form nucleic acids. And those were DNA and RNA. So this is exactly what we're talking about when we are saying we are taking small molecules and building them into larger ones. Now, one thing to note about anabolic reactions is that these reactions require ATP. So we have to put energy in order for these reactions to occur. I sort of remember these reactions as anabolic reactions being used to add things together, and ATP is required. So I just think about the fact that all of these things begin with the letter A. Now, these anabolic reactions are going to be important because they basically allow us to build cells. So in order to have a cell, we need things like carbohydrates, we need nucleic acids, we need lots of proteins. And so these anabolic reactions provide materials for cell maintenance, cellular growth, as well as repair of cells when they get damaged. Now, the name of the type of chemical reaction that is used for anabolism is called a dehydration synthesis reaction. This may seem like a complicated term, but just remember that dehydration means that water is being kicked out. Synthesis, so basically we're removing water. And synthesis tells us that this is a type of reaction in which we're building something. So if we jump down to our image down here at the bottom, we have two monosaccharides that are being hooked together to form a disaccharide. And in order for this to happen, we have a hydrogen and an oxygen that once these two simple sugars are hooked together, a hydrogen and an OH group are being kicked out or being removed, as shown here. And so that is a dehydration reaction. All right, so again, make sure you know that these types of dehydration synthesis reactions are used in anabolic pathways or in anabolic reactions in order to build, to make something bigger. Now, our second type of metabolic reaction is called a catabolic reaction. And catabolic reactions are just the opposite of anabolic reactions. In a catabolic reaction, we are taking those larger molecules and breaking them down into smaller ones. Now, if an anabolic reaction requires the input of energy, a catabolic reaction actually releases energy. So this is a way that we can sort of release energy that can be used by a cell in other ways. So in these catabolic reactions, I always think about the fact that a cat will sort of like, you know how they push stuff off of tables and break things? So I think about cats pushing something off of a table and breaking it apart. And so this makes me think of a catabolic reaction. Now, when we have a catabolic reaction, the type of chemical reaction that's actually occurring is called a hydrolysis reaction. So a hydrolysis reaction is just the opposite of a dehydration synthesis reaction. In a hydrolysis reaction, the two parts of this word are hydro and lysis. So hydro just means water, and lysis means to break apart. So in these hydrolysis reactions, we will be inserting a water molecule, and that is going to insert right here in between two of those individual units. And once that water is added in, we are able to basically break these apart by adding the H and the OH right here. Okay, so I will never ask you to actually draw these chemical reactions, but you should know basically which ones are used to break things down, which ones are used to build things, which ones are associated with anabolic or catabolic reactions. So you should just know the general idea of these. Now, these catabolic reactions are used to basically take proteins and break them down into amino acids. My pen is not cooperating. These catabolic reactions take polysaccharides and break them down into monosaccharides, and they take nucleic acids and break them down into nucleotides. So these are just the opposite of our anabolic reactions. All right, so now that we've mentioned our two types of metabolic reactions, now let's talk about how these reactions can be controlled. Every cell performs both anabolic and catabolic reactions. So every cell needs to be breaking things down and building things. Now, the rates of these catabolic and anabolic reactions have to be really carefully controlled. If we start breaking down everything and not rebuilding anything, you can imagine that the cell would end up in a lot of trouble. On the other side of that, if we start building a bunch of stuff and don't actually break anything down, the cell is going to be just overloaded with these macromolecules. So we have to really carefully balance the rate of anabolic and catabolic reactions. One of the reasons that this balance is so important is because of that ATP that is required in some of the reactions and is released in others. And so it's really important also that we sort of balance the amount of ATP being used with the amount of ATP being produced. Now, if a cell has an imbalance in the number or the rate of catabolic versus anabolic reactions, this can actually damage or even kill a cell. So it can be very dangerous to have an imbalance in these reactions. And different types of cells have different types of specialized metabolic processes. And we'll talk about these kind of specialized processes once we get into individual organ systems later in the semester. Now, if we're talking about metabolic reactions, another important thing to understand are enzymes. And enzymes are actually proteins that catalyze or speed up chemical reactions. So an enzyme is a protein that catalyzes a chemical reaction. And so really we have, or cells have, very specific enzymes that control the rate of all of our metabolic reactions. So how do enzymes actually work? So enzymes are globular proteins, and the way that they actually catalyze or speed up chemical reactions is by lowering the activation energy of these chemical reactions. One important thing to note is that enzymes are not actually consumed in a chemical reaction. So they are used over and over. These are proteins that a cell can sort of keep around and use a bunch of times to speed up a particular chemical reaction. So you can see here in this picture that we have an enzyme shown in purple, and that enzyme is going to be used in order to, or it's going to bind to two substrate molecules. So let's pretend that this is a monosaccharide and this is a monosaccharide, and here we have our enzyme. The enzyme is going to bind to both those monomers and basically hook them together. And so we see this entire polymer that has been produced. The enzyme is coming out of this reaction completely unchanged. It looks exactly the same as it did coming in, which means it can be used again for another chemical reaction. Another important point about enzymes is that each enzyme is going to be very specific to its substrate molecules. So if this enzyme is responsible for speeding up an anabolic reaction and adding these two substrates together, then that same enzyme is not going to be able to hook amino acids together, and it's not going to be able to hook nucleotides together. So these enzyme molecules are very specific to particular substrates. Now, as you can hopefully appreciate from this image, the ability of an enzyme to recognize a substrate depends on the shape of what we call an active site of that enzyme. So the active site is the site where the enzyme is actually recognizing and binding to the substrate molecules. The last point here is just something to sort of help you just with remembering names of things. Enzymes are named a lot of times, not always, but a lot of times, enzymes are named after their substrate with ASE stuck at the end. So the example here is lipase. So lipase is an enzyme that breaks down lipids. And so ASE always tells us that this is an enzyme, and the LIP at the beginning tells us we're talking about a lipid. Some other examples of enzymes that are used to either build or break down certain molecules are things like a protease, and again, we know ASE tells us that this is an enzyme, and proteo tells us that this is an enzyme used to break down proteins. Another example would be, let's see, lactase. And lactase is again an enzyme that is used to break down the disaccharide lactose into two simple sugars. So there are lots and lots of examples, but for right now, just make sure you remember that the suffix ASE tells us we're talking about an enzyme.